When you think of Biobetters, that's actually a relatively new term. So some will be familiar with it, some won't. When we talk about biologics, most people think about antibodies, which are large molecules targeting a specific epitope. We have then generated second generation antibodies, which are the follow-ons to the originator innovator molecules, and these are intended to target the same epitope but have different variable domains, and so their efficacy could be slightly different, hopefully better. We have biosimilars. So biosimilars are molecules which have the same amino acid sequence as the innovator molecules, but they're manufactured differently, so there's potential for them to be slightly different in terms of their efficacy. A lot of the original efforts were toward our molecules such as rituximab. So this is targeting anti-CD20, and the original biosimilar efforts were coming up with the next generation in terms of the biosimilar for anti-CD20 and the rituximab, looking at the same amino acid sequence. How can we manufacture these different but make them more powerful than the original, or at least equal in their potency? Then we have Biobetters. So this is where you have the same epitope that you're targeting, but you're improving the properties. So that means you potentially will have lower dosage, less frequency in terms of the dosage. You're targeting the same epitope, but you've improved the biophysical characteristics of the molecule so that it can be more potent in a variety of different ways. For the Biobetters and Next Generation Biologics Conference, I selected some of the greatest minds in the field, but they were not just the greatest minds in the field, they were also selected for what they're bringing to this conference, which is we have Amy Rosenberg, who was trained as a clinician. She's currently at the FDA, so she's very familiar with the regulatory process. We have Amy Cleon, who is at the NIH, at NIAID. She is a clinician, she's also a researcher, so she's very familiar with what happens in clinical trials. And we also have Sakdev Sidhu. He's at the University of Toronto, so he brings not only a bioengineering perspective, but also an academia perspective. And then myself, being in industry at Biogen, is very familiar with the drug development process. And so each of us will be identifying individuals from our own sectors, bringing them together into a cohesive meeting that will allow us to address the most complex scientific questions. When I think about who should be attending this meeting, the best answer is everyone. Anyone who's interested in where the field is going. What are the next therapeutics that need to be developed? What are the criteria for those therapeutics that we need to be thinking about? That's what's going to be important. What are the regulatory hurdles that we need to be considering? Anyone who's interested in the drug development process, thinks that they have a good idea for something that could eventually become a drug, these individuals should really come to this meeting, because we're going to be talking about it from all of the different perspectives, and we're going to be thinking about the most complex scientific questions, why they're important, what are the factors that need to be considered. In terms of moving the field forward, it's the different perspectives. So if you consider the fact that we have individuals who are in the regulatory space, you have individuals who are in academia, so they'll be coming up with the next ideas of what biologics we need to be considering, what are the targets we need to be considering. You have individuals who are in government and who are thinking about what are the clinical trials that we need to be putting together in order to make sure that these targets make the most sense. That is the aim of the conference. That is the goal of the conference, and we believe that its uniqueness of bringing in all of these different minds who have the different perspectives will actually make it a better conference. In terms of my Keystone Symposia experience, it's actually been a very transformative process. When I was first introduced to Keystone as a graduate student, I wasn't really familiar with what these meetings meant. I had no real appreciation for what a smaller meeting meant, but I learned that it's the intimacy of these meetings that makes them so great. The opportunities to speak with everyone one-on-one, if you choose, or in groups, if you choose, or to toss around ideas and brainstorm, that's critical. That's as an attendee. Then when you become a speaker, it's a different show. It's a different criteria, it's a different mindset, because your role becomes telling the story, making sure the story that you're telling makes sense for the topic that is the focus of that meeting. And that is just as important as attending and absorbing the information. And then as a fellow, a Keystone Symposia fellow, this was the next level in my own development. So this was understanding what the conference development process is, taking that information into account, and really thinking strategically. When I was selected as an organizer, this was very important because I knew all of the different components of the development process that were important for Keystone Symposia. And I thought about those. I thought very strategically, what makes the most sense? What is going to move the field forward? What do we need to be considering? Who are we inviting? Who are we having to give talks? Who are we missing? And how broad should it be? How narrow should it be? These were all critical aspects that we had to consider. But when you think about the Keystone Symposia conference that we've put together, as I mentioned, it's a bringing together of different perspectives. Individuals who are bioengineers, individuals who are clinicians and working with patients, individuals who are at the bench, individuals who are regulators. So I don't believe that anyone would argue that bringing all of these minds together is very important for moving the field forward.